nature of variations. Darwin's theory was based on the idea that natural selection acts upon small inheritable changes. Very small inheritable changes means these changes are inherited to their offspring, transmitted to their offspring or next progeny. So these are small changes. These small changes are denoted as continuous variations. Denoted as continuous variations. According to Galton, there are abrupt changes, means large differences, abrupt changes which are sufficiently different from each other. Which are sufficiently different from each other. He called these changes discontinuous variation. He called these changes discontinuous variation, which are much important in evolution. So these are discontinuous variation. Now, so suppose say for example, the differences when occur inheritable changes, suppose there is 1, then it becomes a 2, it becomes a 3, like that. Small changes, inheritable changes. If 1 changes into suppose 20, so there is abrupt change, that is a discontinuous variation, and small inheritable changes are continuous variation according to Darwin. Then individual variations. Individual variations. The differences among individuals of same population are called individual variations. From evolution point of view, these may be non-genetic or genetic, means there may be variation in the genetic or non-genetic, no variation, no change in the genetic material. Phenotypic modifications that do not involve genetic changes. There may be phenotypic variations, but that do not involve genetic changes are known as non-genetic individual variations. Non-genetic individual variations. These include age variations, because as per age variations occur, age variation, seasonal variation, in different seasons different phenotypes appear, and habitat variations, means where they live, habitat variations. These are phynogenetic or phenotypic non-genetic individual variations. Individual variations but non-genetic, no genetic variation. On the other hand, no two individuals of same species or population are genetically identical. No two individuals of same species or population are genetically identical except monozygotic twins. Twin, monozygotic twin, that means from one zygote if the twins are formed, then they are known as monozygotic twins and they are identical. One is the Xerox copy of another, identical. So except that, no two individuals are exactly alike. These genetic differences are expressed as phenotypic variations. These genetic differences are expressed as phenotypic variations. Then geographic variations. geographic variations. Occurrence of differences among spatially segregated populations of a species is called geographic variation. If they are spatially segregated, they live in different geographical regions, there are variations. These are known as geographic variations. These are mostly adaptive, means they adjust to their particular environment. So these are mostly adaptive and are inevitable consequences of geographical variations of the environment. So that is because of geographical variations of the environment, because the environment is different in different geographical areas. For example, dark skin of human race, dark skin of human race in the tropics is advantageous. Tropics रे जो ह्यूमन पॉपुलेशन रोची तांकर स्किन टा पूरा पूरी डार्क, that is advantageous. कौन पाई? So dark skin of human race in the tropics is advantageous because it shields the underlying tissues from sunburn. Sunburn रू रक्षा करिया पई ताकू से अंडरलाइन टिश्यू को सील करूची, कवर करिकी रखुची from sunburn. But in higher latitudes where sunburn is no threat, जोटी sunburn मोटे threat नुह, but in higher latitudes where sunburn is no threat, translucent skin is an advantage. Translucent skin is an advantage because such skin allow penetration of UV components, ultraviolet components of sunlight into tissue. It allow penetration of ultraviolet components of sunlight into tissue because this is necessary for vitamin D synthesis. So that is an advantage in higher latitudes, the population of higher latitudes. But in tropics, the skin is dark, that shields the underlying tissues from sunburn. Here it is translucent. It allows the UV components to enter because it is necessary for vitamin D synthesis. Races of human are Africans, Orientals, Caucasians, etc. are products of geographic variations, Then sources of variation. Sources of variation. Inheritable variations may arise through number 1, mutation, number 2, genetic recombination. So, inheritable variations, those variations which are transmitted into their offspring, these are known as inheritable variations. So, these may arise through these processes. Number 1 is mutation, number 2 is genetic recombination. First, we will discuss about mutation, then we will go to genetic recombination. So, mutation. Mutation literally means change. Any inheritable change in the genetic material is called mutation. Any inheritable change in the genetic material is called mutation. A mutation occurring in somatic cells, means body cells, somatic cells, is called somatic mutation, which is also called non-genetic, because these genes are not transmitted to their offsprings. Because in the body, the cells can be divided into two categories: somatic cells and germinal cells or germline cells. Germline cells are present in the gonads, testes and ovary, where the gametes are formed, the sperm and egg are formed. So through them, these genetic changes are transmitted. So in the somatic cells, the mutations which occur, these are non-genetic. Mutations occurring in the germline cells is called germinal mutation, which is heritable through the gametes to the progeny, through the gametes to the progeny. Mutations can occur spontaneously or can be induced. It occurs spontaneously or it can be induced. Mutation is a forward as well as a backward phenomenon. Suppose A1 mutation occurs, it becomes A2. A2 again mutate back to A1. So it is a forward as well as a backward phenomenon. First, gene mutation. We will discuss gene mutation. Gene mutation. Gene mutation is defined as changes at DNA level, because the chemical composition of gene is the DNA. So at DNA level is defined as changes at DNA level which affect nucleotide structures of a gene. So nucleotide structure is affected here. Hence, it is also called point mutation. Gene mutation is also called point mutation. Point mutations may occur by base substitution, because in the nucleotide, you know, DNA is a double nucleotide chain, and here each nucleotide possesses nitrogen bases. So point mutations may occur by base substitution. If base is substituted by another base substitution, where one base pair is replaced by a different one. One base pair is replaced by a different one. As you know, in the DNA, the base compositions are A pairs with T combines with T, G combines with C. So ATGC, adenine, guanine, cytosine and thymine. So these nitrogen bases, adenine, guanine, are known as purine bases, and thymine, cytosine, these are pyrimidine bases. So base substitution in DNA may occur in 12 ways. In 12 ways, base substitution. So now from this diagram you can see A may be substituted by G, G may be substituted by A, T may be substituted by C, C may be substituted by T, A and T there will be substitution, T and G there will be substitution, like that. So all total you see 12 ways, 1, 2, 3, 4. 2, 2, 2, 2. So total 8 plus 4, 12. 12 ways base substitution occurs. So when replacement occurs between two purines or two pyrimidines, it is called transition. So these are transition. Between two purines, A is substituted by G or G is substituted by A. Similarly, T is substituted by C, C is substituted by T. So these are known as transitions. And if it is between purine and pyrimidine, this is known as transversion. This is known as transversion. So there are 12 ways. Addition or deletion of one or more nucleotide may change the reading frame. If one base or more are added in the nucleotide, addition or deletion of one or more nucleotide may change the reading frame of codon from the site of change onwards. जो जगार गोटे पर्टिकुलर पॉइंट रे जदि गोटे कोडन आड हुए, कोडन न्यूक्लियोटाइड बा आउ अधिका आड हैला, ताले एंटायर फ्रेम विल बी चेंज्ड. एंटायर फ्रेम सेटा को कहा जाउची फ्रेम शिफ्ट म्यूटेशन. सच चेंजेस आर कॉल्ड फ्रेम शिफ्ट म्यूटेशन. ए सब्स्टिट्यूशन म्यूटेशन changes that codon only. तेणु दुई प्रकार आम ये कहले गोटे है सब्स्टिट्यूशन म्यूटेशन आ गोटे फ्रेम शिफ्ट म्यूटेशन. सब्स्टिट्यूशन म्यूटेशन चेंजेस दैट कोडन ओनली. से पर्टिकुलर कोडन आम जो ट्रिपलेट कोडन रही तीनटा नाइट्रोजन बेस दे रिप्रेजेंट वन अमीनो एसिड, से गोटे पर्टिकुलर टाको चेंज करिवो, आउ किछि नुह. सो ए सब्स्टिट्यूशन म्यूटेशन चेंजेस दैट कोडन ओनली व्हेयर द सब्स्टिट्यूशन ऑकर्स. बट a frame shift mutation changes the sequence of codons. Sequence of codons. Now you can see here So you can see here the combination was AT. Now during replication they isolate the separated, then synthesis the other bases or co-nucleotides combine here, and here you can see there is the tautomeric shift of pairs. So here A must pair with T and T with A, but here instead of T it pairs with C. Then again replication occurs, both are separated and new nucleotides are added. So C now combines normally with G. So you can see AT is replaced by GC. It is substitution mutation. So AT becomes GC. So A goes tautomeric shift here. A goes tautomeric shift and pairs with C and pairs with C. Here A pairs with C instead of T, that is known as tautomeric shift. Now the frame shift mutation, entire frame is changed. TAC, TCC, AAC, ACG, ATA. Now from this RNA is synthesized. A, U, G, A, G, G, U, U, G, U, G, C, U, A, U. Now the protein which is synthesized contains methionine, arginine, leucine, cysteine, G insertion. Now DNA, TAC, TCC, AAG, CAC, GAT. Then RNA, AUG, AGG, then UUC, then GUG, then CUA, and the protein methionine, arginine, then valine, leucine, so now you can see here, this is the DNA having the codons are TAC, because triplet codon TCC, AAC, ACG, ATA. So the normal sequence example. Now from this RNA is synthesized. See, thymine replaced by A, pairs with A. Then from A, instead of thymine, you know, in RNA uracil is present. So U, C with G, from T, A, C, G, C, G, then UUG, then UGC, then UAU, and these are the amino acids of that particular protein. Now see, if G is inserted here, now particularly this point, so G is inserted, now it will become AAG, then CAC, then GAT, एणिक सीक्वेंसटा पूरा बदली गला। ये जो ट्रिपलेट कोडन थिला एगुड़ाक सब चेंज हो गला। सिमिलरली आफ्टर आरएनए यू सी तीनटा अमीनो एसिड बा पर अमीनो एसिड जो गुड़ाक से गुड़ाक मध्य चेंज हो गला। सो एंटायर प्रोटीन इज चेंज्ड। सो phenotypic variation will occur. Mutations can occur spontaneously or can be induced. Mutation can occur spontaneously or can be induced. Now, example of a point mutation. One example of point mutation is GTG, CAC, CTG, ACT, CCT, GTG, GTG. This is the 3' end, this is the 5' end of the DNA, then 3' end, last one here, this is 1, 2, 3, 4, 5, 6. Sixth one we will consider here CAC. CAC. This is the 5' end because the two nucleotide chains, one chain 5', others is 3' end. So 5' end and 3' end. This is the normal. This is the point mutation DNA. Now normal one, in normal one the GAG is the 6th one. Now CTC, then GAG, 6th one, and glutamic acid. This is the 6th amino acid, and here mRNA, messenger RNA, GUG, the 3' end. Now it is the 6th, then amino acid is valine. 6th amino acid is valine. This is sickle cell anemia. Sickle cell anemia. So you can see here, this is the normal sequence, 5' end and 3' end. So GAG normal, and here it is CTC. But here you see point mutation, instead of GAG it is GTG. A has been replaced or substituted by T. Now mRNA becomes GUG, and the amino acid which is produced by this is 6th amino acid, that is valine. But here in normal it is glutamic acid. So in the hemoglobin there is change in the amino acid, 6th amino acid. So in sickle cell anemia there is a single amino acid substitution. in sickle cell anemia, there is a single amino acid substitution in the beta chain of hemoglobin. Alpha chain, beta chain are there. So in the beta chain, single amino acid is changed. That is the sixth one, and it is the glutamic acid is replaced by valine. So valine instead of normal glutamic acid at position 6, and that becomes sickle cell anemia. The red cells become sickle shaped, sickle cell anemia. Then chromosomal mutation. Chromosomal mutation. Chromosomal mutations are large scale changes involving more than one gene, because the entire chromosome contains genes. So if there is any change in the chromosome, then there will be change in the genetic material. So it involves more than one gene. These mutations are classified as number 1, chromosomal aberrations. Means structural changes in chromosome, structural changes in chromosome, and under this A, changes in the number of genes, changes in the number of genes. B, duplication. A, now under this, changes in the number of genes, it is either deletion or deficiency. Then duplication. Then B, changes in the arrangement of genes, changes in the arrangement of genes, inversion, and translocation. Then number 2, numerical changes. Means changes in the number, numerical changes in chromosomes. A, fusion and fission. Fusion and fission. B, aneuploidy, and C, euploidy. So these are the chromosomal mutations. So it can be divided into 2 categories. First is structural changes, that is known as chromosomal aberration, then numerical changes. Under structural changes, changes in the number of genes and changes in the arrangement of genes. Same genes are present, but arrangement is changed. Now changes in the number of genes includes deletion or deficiency and duplication. Similarly, changes in the arrangement of genes, inversion and translocation. Then numerical changes means changes in the number of chromosomes, fusion and fission, then aneuploidy and euploidy. So these are the chromosomal mutations. Now let us discuss one by one. First is deletion. See, deletion or deficiency. Deletion means loss of chromosomal material. Chromosomal material is lost. It may be terminal, that is from the end, or intercalary involving two breaks. Now see, this is terminal because here a break occurs and this is lost. Now the chromosome becomes 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 is lost. So this is terminal. Terminal. Now if from the middle portion, that means two breaks are involved. These are the two breaks and these 2, 3 are deleted. So it becomes 1, 4, 5, this is the centromere, 6, 7, 8, 9, 10. This is intercalary. Intercalary. So deletion. Now let us see what is the phenotypic effect of deletion. The phenotypic effect depends upon the amount of genetic material lost. How much genetic material is lost, ता ऊपर depend करूची phenotypic effect. Example, a deficiency on the short arm of chromosome number 5 in human. A deficiency on the short arm of chromosome number 5 results in the XII chart. Results in the XII chart syndrome in man. XII chart means cat-like cry. They cry like cats. Cat-like cry. That is XII chart. Then duplication. Duplication. It refers to the presence of a chromosomal segment more than once in a set of chromosomes. So presence of a chromosomal segment more than once. So every segment is present once. If it is present more than once in a set of chromosomes, it is called duplication. Example is bar I in Drosophila. Bar I becomes bar shaped, not rounded. Bar I in Drosophila. Duplication of 16A regions in the X chromosome. Suppose this is the X chromosome and this is the 16A region. This is the X chromosome, 16A region. It is present once. So duplication of 16A region. Now if another 16A region is present here, so duplication occurs. Duplication of 16A region in the X chromosome results in the narrowing of the eye. Eye becomes narrow. Eye with less facets. 3 regions of 16A results in ultra bar. 3 regions of 16A results in ultra bar. So 2 regions means bar, 16 one single region, that is normal eye, it is bar, and if three sets are present, three regions are present, then it is ultra bar. So normal I like this, then bar I becomes this, and ultra bar is like this. The number of facets are reduced. So normal I, bar, ultra bar, that is duplication. Then inversion. Inversion. The gene order of a portion of a chromosome is reversed due to the rotation of a segment through 180 degree. The rotation of segment through 180 degree. That means, suppose this is the chromosome, and here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Suppose here there is a break. Here there is another break, and this segment rotates through 180 degree. Then the arrangement, and it will join fuses 1, 2, 5, 4, 3, 6, 7, 8, 9, 10, like this. So now you can see here this region is reversed, rotated. So it involves two breaks, and then it rotates through 180 degree, and again it joins. That is known as inversion. In inversion heterozygote, a loop is formed during synapsis. Synapsis, as you know, in prophase 1, the homologous chromosomes, they pair with each other, and how they will pair? Now, this one and this one, they will pair. How? Because like this, and this is the normal. This is the loop. So 1, 2, 3, 4, 5, 6, like this, 7, 8, 9, 10. See, this is the normal arrangement of one chromosome, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Now, if we will go to this one, 1, 2, then 5, 4, 3, 6, 7, 8, 9, 10. So, a loop is formed during synapsis. This is the loop. During synapsis, a loop is formed. If the inverted segment includes the centromere, it is called pericentric inversion. If the centromere is present here, then we will say this is pericentric inversion. And here the centromere is not present in the inverted segment, so this is paracentric inversion. So pericentric and paracentric inversion. Inversion suppresses the crossing over. Crossing over is suppressed by the inversion. Inversion may change the appearance, that is from metacentric to submetacentric or acrocentric chromosome. So suppose this is the chromosome. Now here it is the centromere. So it is not metacentric, it is submetacentric. Suppose here there is a break and here there is a break. Now it will rotate. Then the structure will be like this: 1, 2, 3, suppose 4, 5. It will break tahla. Now 1, 2, 6, 5, 4, 3, 2. ଏଠି ଗୋଟେ ବ୍ରେକ୍, ତେଣୁ ଏଇ ପୋର୍ସନଟା ଦେଖ, ତାପରେ 6, 7 like this. ତେଣୁ ଏଇଟା ହେଲା submetacentric, ଇଏ ହେଇଗଲା metacentric chromosome. That may be also telocentric or acrocentric. Inversion may not have any drastic effect. But all the genes are present, only arrangement has been changed. But there may be some effect known as position effect. There may be some effect known as position effect. Then translocation. translocation. This one, it involves a transfer of a chromosomal segment to a non-homologous one. A segment of one chromosome is exchanged by or with a non-homologous chromosome. Either it is shifted to that non-homologous chromosome or it is exchanged. It may be simple or reciprocal, means a portion is exchanged between two non-homologous chromosomes, that is reciprocal. Translocation heterozygotes may produce gametes with duplications and deletions. Then fusion and fission. Fusion and fission. Fusion means two chromosomes will fuse. Fission means one chromosome will break. Fusion and fission. So this is, you can see, fusion. So two chromosomes are there. Here there is a break, here there is another break. Now these two, they fuse with each other and the centromere remains in the middle, so it becomes metacentric. It is fusion. So fusion means the union of two non-homologous chromosomes into one. Two non-homologous chromosomes, you can see they are non-homologous chromosomes, fusion occurs, and in fission, one chromosome splits into two. One chromosome splits into two. Suppose this is one chromosome, and it splits into two, that is fission. So one chromosome here, this is the centromere. Now centromere break here, there is a break in the centromere, so they are separated, that is fission. So one chromosome is converted to two chromosomes. So fission, so fusion reduces the chromosome number. Fusion reduces the chromosome number, whereas fission increases the chromosome number. You can see here the fusion, two chromosomes, they become one. Here in the fission, one chromosome becomes two. So it reduces the chromosome number, it increases the chromosome number. These are known as Robertsonian changes. These are known as Robertsonian changes. These are known as Robertsonian changes. It is regarded that larger chromosomes are derived by successive fusion of small chromosomes. যেতে বড় বড় ক্রোমোজোম সবু রয়েছে সেগুড়াক ছোট ছোট ক্রোমোজোমর ফিউজন হয়েকি ফর্মেশন হয়েছি। অল গ্রেট এপস, বড় বড় এপ, ফর এক্সাম্পল the chimpanzee, gorilla, orangutan, all great apes such as chimpanzee, gorilla, orangutan have 2n is equal to 48. They have 2n, two sets of chromosomes. Total chromosome number is 48. 2n is equal to 48, where man has 2n is equal to 46. Here these are the great apes. Great apes, they have 2n is equal to 48, and in man 2n is equal to 46. It is most likely that man has descended from any one of these. Man has descended, as per evolution theory, man has descended from any one of these 48 stock through centric fusion at some stage. Ko gote stage re centric fusion heiki number has been reduced to 46. Instead of 48, that has become 46. Then aneuploidy, the number aneuploidy, changes in chromosome number within the set is called aneuploid. Within the set, changes in chromosome number is known as aneuploid. Addition to normal set is called hyperploidy, and loss is called hypoploidy. Addition to the normal set is called hyperploidy, and loss is called hypoploidy. Hyperploidy are classified as as 2n plus 1, that is trisomic, 2n plus 1 double trisomic, 2n plus 2 tetrasomic, etc. These are hyperploidy. Now see, suppose the normal chromosome set are AA, BB, CC. Suppose here 2n is equal to 6. Now 2n plus 1 is added here. So if it becomes AAA, then BBCC, it is called as a trisomic, because instead of one pair, 3 chromosomes are present, same chromosomes are present, the number is 3, it is trisomic. Then double trisomic means A in triplicate, B in triplicate, C. So here plus 1, here plus 1, so double trisomic. Now you must say 2n plus 1 jaha 2n plus 2 say no, 2n plus 2 will be here, see. So instead of 2, 4 same chromosomes are present, so 2n plus 2, this is 2n plus 1, it is known as tetrasomic, charita chromosome gotakar. ता डबल ट्राइजोमिक दीटा जगह 3-3टा रोची एठी रहिला हमरो नॉर्मल सेट, सो दैट इज hyperploidy. Then hypoploidy, hypo means loss, hypoploidy, 2n minus 1, that is monozomic. 2n minus 2, that is nullizomic, etc. Now see, this is the normal. Now 2n minus 1. So here let us say minus 1 means A is lost, then AA, BB, CC. It becomes 2n minus 1. That's why it is, as because it is only 1, then it is called as monozomic. In one pair, a single is present. Monozomic. যেମିତି তিনটা রইলা আমি কোচে ট্রাইজোমিক, চারটা রইলে টেট্রাজোমিক, এটি রইলা মনোজোমিক, দুইটা হলা নর্মাল। দেন 2n minus 2, it is 0, then BB, CC. ता माने गोटा करे किछि आउ नाही, नील रु हैला nullizomic. One pair is totally lost, nullizomic. So you must say 2n minus 1, that is double monozomic, we can say A, B, C, C, like that, so on. So these are hypoploidy. Then euploidy. So that is aneuploidy, then euploidy. Change in the number of sets of chromosomes from normal diploid condition is called euploidy. Change in the number of sets of chromosomes. Normal is 2 sets are present, 2n, and there may be change in the number of sets. Either it may be 1n, or it may be 3n, or 4n, 5n, 5 sets, like that. If single set is present, it is called haploidy. 2 sets diploid, that is the normal. If 3 sets are present, triploid. 4 sets tetraploid, 5 sets pentaploid, and so on. So change in the number of sets of chromosomes from normal diploid condition is called euploidy. When the organism contains only one set of chromosomes, single set, one set of chromosomes, that is n, the condition is called haploidy. The condition is called haploidy because normal 2 sets are present. Now half of 2 is 1, so 1 set is present, that's why it is called haploidy, not monoploid. Haploidy. Example, the drone in honeybee, mohu machiro male ta taku kuajae drone. Drone in honeybee, these develop from unfertilized egg. Karan egg re haploid set present, n chromosome present, they are haploid because in meiosis chromosome number is reduced to half. Egg re roila n, 1 set, sperm re roila 1 set. If they fuse, they will form zygote, that becomes a 2n. किंतु एठी कौन होची ना egg रू without fertilization development होची, that becomes the drone. So drone becomes haploid. So these develop from unfertilized egg, that is known as parthenogenesis. Parthenogenesis, that is development without fertilization. So example is the drone in honeybee, mahumachiro drone in honeybee, that is haploid. If organisms contain more than 2 sets, if organisms contain more than 2 sets, it is called polyploidy. It is called polyploidy. It may be triploid, 3n, tetraploid, 4n, pentaploid, 5n, hexaploid, 6n, and so on. Polyploidy may originate within the species. If within the species the polyploid is obtained or originate, it is called autopolyploidy. Same species भितरे जदि formation हला polyploidy, ताले ताकु कुआ जाउची autopolyploidy. And when two different species contribute to polyploidy, it is called allopolyploidy or amphiploidy. Amphiploidy. So, if within one species it is obtained from one species, it is called autopolyploidy, and two different species if contribute, it is called allopolyploidy or amphiploidy. Example is the common eating banana is triploid. Common eating banana, जो कदली आमे खाऊचे common eating banana से गछ, that is triploid. One interesting thing, जदि आमर जेतबेले मेयोसिस होउची होमोलोगस क्रोमोसोम पेयर करूचंती दीटा थिले पेयर हबो, जदि तीनटा अछि तीनटा भितर पेयरिंग हे परिबोनी, तेनु गैमेट फॉर्मेशन हबोनी, तेनु कदली गछरे आमर फूलरू मंजी हुए नाही कि मंजीरू गछ होबार संभावना नाही, नो ये गछ पुआरू ही गछ होउची ता मूलरू, सेथि पई बिकॉज इट इज ट्रिप्लॉइड, तीनटा सेट अछि पेयरिंग हे परिबो नाही. सो अनदर एग्जांपल इज राफानो ब्रासिका. Raphano brassica, where 4n is equal to 48. 4 sets are present. 2n contributed by Raphanas and other 2 sets by brassica. It becomes 4n is equal to 48. Then recombination. Class number question. Nebe. Oh, okay, more. Recombination. Recombination is the process of mixing of available genes into varieties of genotypes. जेते प्रकार रो जीन सब रही तार मिक्सिंग है कि वेराइटी ऑफ जीनोटाइप फॉर्मेशन हबो. It is also a primary source of variation and no less significant than mutation. Mutation जेतिक significant, ये मध्य सेतिकी significant, not less than mutation, not less significant than mutation. Recombination of genetic materials arise by 3 ways. By 3 ways, recombination of genetic materials occur. That is, random assortment of genetic material, random assortment of genetic material, then genetic exchange during crossing over, and crossing two different species or hybridization. So, recombination of genetic materials can arise by 3 ways. These are random assortment of genetic material, then genetic exchange during crossing over, then crossing two different species or hybridization.